years ago, I mean, before the advent of microcomputers, there weren't any easy metaphors for what the brain did. As you were growing up, did you have any notion of what the brain actually was?
Not a lot, but I will say when I was, I guess I was in junior high school, I entered a little contest and made a little poster, use your brain, not your accident policy. It's the first money I ever earned was by myself, was winning that. So, but yeah, no, I mean, your major point is that we've known nothing about it. It still is a mystery, right, despite all these methods. But when I started out, we basically were in awe of all the big questions. You know, how do you see? How do you feel? You know, what's the nature of thought? Is consciousness important, and are we the only ones who have it? Right? All of those questions. And so the field, as you know, it was in a certain sense disciplined. It's too early to look at all those questions. You know, theory of mind didn't exist in neuroscience. But it was okay to look at sensation. It was okay to look at movement. And people had worked out a lot about the spinal cord and reflexes and so on. But I just hit coming through here and going, you know, being born and all, at a time when everything changed. It just, so technique after technique opened up. And you know, concept of the brain, I think now a lot of people have at least a rough idea that, you know, there's electricity in there and lots of transmitters and chemicals and imaging the brain. I mean, everyone would agree is what put it in the New York Times and, you know, put it everywhere in front of everyone. More and more people have their brains imaged for one reason or another. So it's really part of the whole of biology has opened up to people now, at least in developed countries.
And in your time of study, in your lifetime, I mean, from the moment you were a child and began thinking about these sorts of things, the brain has gone from utterly opaque to the first hints of actually understanding the real structure. Mm-hmm. And it's an endeavor that involves more than just one science.
Oh, yeah. Yeah, very much so. And so I don't know whether we're going to talk more about, you know, MIT and this very subject, but MIT has been so special that way because early on, even before the kind of biologic, it's sort of like the Greeks, you know, they were pretty good in chemistry and physics, but they didn't really do a lot of biology. And MIT was outstanding in biology up to the level of the cell. The biologists were not working on the brain. So, but what we had, I mean, we had Norbert Wiener, we had, you know, early artificial intelligence, we had all the different people who were basically using the human intellect to try to figure out how it might work and lay some kind of computational restrictions on what we might look for. And so what's happened is that interestingly, MIT's in a very good situation now because biology is now very interested in brain. Many different disciplines have come to the work on the brain. And the computational people are more and more working with experimentalists. And the physicists who were the heart of MIT in a lot of ways, along with mathematicians and engineers, of course, they now are interested in the brain. So it's a very curious time. It's a very interesting time when some people think the way to tackle it is still to look at the, you know, what would be the smallest computational unit, and then we'll go find it. So they're starting out with their ideas and their concept and whatever brutal strength they've got as, you know, their own brains. And then the other part are more kind of Darwinian people. Let's look, see what nature did, and then apply those computational tools. And so there is a certain, there's a wonderful kind of, it's not a tension, it's just a back and forth communication between those two different sets of ideas. And that is MIT now in neuroscience. And MIT was poised to do that. And there are a few other places where this sort of thing goes on, but there aren't a large number. So,
how did it occur to you as a young girl that you could have a career in science at the time that you grew up?
My dad was a scientist and a physician. My mother would have been a physician. It was tough in those days for women, especially she was from Northern Ireland. But they were of a medical family. And my father loved science. He loved medicine. He was very good at it. But he loved science. It was evident. And so I kind of poked around in there, and I, quote, worked with him. He was nice and let a kid help him.
Where? In what parts of his practice?
Oh, no, not in the practice, but just when he was, for example, writing papers or something. And so we talked a lot about things like that. And they both loved nature, which I think is, it's fundamentally a part of why I do what I do. And I happen to think the brain is a mighty fine product of nature. So it was, for me, kind of in a way easy, except there was this, female thing. So I grew up in redneck, northern Florida, Panhandle, 13 miles from the Alabama border. Little girls didn't do things like that, right? And so, among others, we couldn't take science. That was too bad. So.
What grade are we talking?
9th grade. That was the first grade of science.
And you couldn't take science?
I wasn't allowed to, no. So we took home economics, and I sort of learned to sew. But then my parents had to.
You've done a great job.
Thank you very much.
Stunning. Yeah.
My parents had to kind of ship us off to boarding schools. I have a brother who is a physician.
How did your parents explain to you this restriction on science in 9th grade? What did they?
Oh, are you too young to know? I mean, it was sort of, remember, this is the Deep South, so it's so different from now, it's almost hard to describe, which is why I try to, anyway, help girls to this day, but not by talking about the way it was, but just by helping them. But anyway, the way it was that women did one thing and men did another thing, basically, and it wasn't really, my parents weren't happy. I was sorry, but I wasn't a science wonk. I mean, I was just a student. But, if people had parties, the women talked in one place and the men talked in another, and the women took you to piano lessons or, worked in the kitchen, and the men went out. So, how can I put it? It took a long time, a really long time in my case, to kind of feel it's okay, right? See, I was the only, it's funny, I was the only female faculty member in my building for many, many years. Once we moved to a building called E25 or not, and we've moved out of it into this glorious science complex. But there was more to the department. It was in another building, but it was curious, it's 6 stories. So when I was at Harvard as an undergraduate, I realized you made me think about these things. I hadn't really thought of them, but I never saw a female professor. I knew one woman, turned out she was an instructor. I never saw a female professor. I heard there was one in astronomy. So it was a different time,
Yet, how does that, how does that shape your discipline as a scientist? I mean, you're the only woman in the room, you're the only woman in the department, you're the only woman in the class,
Yeah. Right. So like well, it shaped things in certain ways. Like women were not allowed to go to the main library at Harvard, Widener. We were assigned seats. So like when we took organic chemistry, we were assigned seats in the front rows. So that was a good influence. We were way up front. It was easy to see. But again, it was the way, how can I say this? There came upon all of us and fortunately I was part of it, a time when it was okay to protest almost everything. And I lived right smack in the middle of that. When I went to Harvard Radcliffe, then Radcliffe, we all had 31 o'clocks, so we had to be in by 1 o'clock. We were allowed to stay out till 1 o'clock 30 times in a semester. By the time I graduated, we were not allowed to go out after 10 o'clock.
After 10, but you got...
So you signed out and you signed in? You don't know that?
And you had to...
when a girl went out, like after dinner or something, she signed out, like going to the library or wherever, going out of a dormitory.
That I understand.
The girl had to be in by 10 except 30 times.
So you got 30 times to go wild between 10 and 1.
You got it, right. And so by the time I graduated, there were no rules at all. None. So you can imagine people did go wild. I mean, it was a very crazy time. Right?
Just
so
on the 31 o'clocks, just for 1 second, did you, some evenings, kind of compare notes with other women friends? Oh, I've got 3 to go. Oh, I've got 6 to go.
Oh, no.
No.
Oh, no.
Did everybody know how close they were to the 30, or was it just one of these?
I'm sure I must have gotten in trouble. I can't imagine I only went out for 30 times. But no, but MIT did. The first time I heard about MIT, it was really awful. They had this place called Bells, which is where you signed out, and where phone messages were received. It's just such a different era. And the MIT guys would come around on Saturdays and call up on Bells to anyone. Anybody want to go out? So we had a very low opinion of MIT in those days, I'm sorry. I did marry an MIT undergrad, so, but.
Although, you know, experimentally and statistically, maybe that was a better way of solving the problem. You just go to where the women are and call on the phone and see if someone will come down.
Now I think it's fine.
Elegant solution.
Exactly, right,
Yeah.
as usual.
You said something really profound a moment ago. There came a time when it became okay to protest almost everything.
That's right.
How did that moment arrive?
Do you mean in our country, in the world?
No, in just your community of, I mean, I've heard the story from political protesters' point of view, from president's point of view, but from the point of view of a young scientist.
Okay, so now, all right, so now, like, President Johnson, for example, was running this place during the Cambodian crisis and all this, Vietnam, the whole thing. Or not the whole thing, but part of it. And, you know, Harvard was almost in flames, and somehow he managed to keep the lid on at MIT, but I've always admired him for that, but I think a lot of people at MIT had the sense that it probably is important that some people just keep, you know, going on and doing the thing that they were doing, but there was enormous tension. I mean, I remember very vividly being a student. In those days, I used a microscope a lot, and sitting at my microscope at night, and you know, a guy coming in and saying, "How dare you work? You know, how dare you do this?" And so I thought about it a huge amount. We talked about it endlessly. When I was in college, earlier than that, we were sure we were going to be blown up by the Soviet Union. We were absolutely sure. We just sat around and talked about, you know, when will that happen? So, but the protest, I mean, it was all through this country and many other countries. It just kind of boiled up, didn't it, after all those wars and people getting a bit fed up with it. So I did not go to Alabama, you know, to work with the blacks, but I did things other ways. I would say I was quietly active, but I've never gone out and, you know, burned my bra for women or something like that, because I think there are ways to be quite effective as a Protestant without doing that.
So in this period of time, relationships between men and women are in play, socially everything is in play, scientifically were things in play that as a young scientist you began to see things in ways that were...
You know, I think I was saying that this thing about big questions, honestly, there was a sense of, or at least certainly with me, and I think with many of my colleagues who were very young, a kind of a sense of reverence about, how could the brain guide behavior and mind, brain, those questions, they were looked at with a lot of respect. And so part of the freedom that came was one method after another opened up. Suddenly, Hubel and Wiesel were recording with microelectrodes. Jerry Lettvin was here. And they were finding cells that seem to be related to perception, to actually how you see. This was somehow captivating. This was beyond a reflex in the spinal cord. And then, well, chemicals. Well, they had been, there are some chemicals, right? I mean, I love that because I've always loved chemistry, and it influenced me a lot. But suddenly, you could say, well, no, this is a neurotransmitter, and the way a synapse works is this stuff is spewed out, and if you have too little, you might get Parkinson's disease, right? So again, in the same time, there was a sense of empowerment that bit by bit came. And I think probably another important thing is that, how can I put it? I'll put it very directly. We were coached almost, just short of being coached, that pure science is the kind of science to do. Don't think about an application, Don't think about the consequence. You have to think about, if it's not pure mathematics, at least it's with your whole being trying to explore some problem in nature, physical nature. And, well, bit by bit it turned out that, gee, one thing after another might have a heck of a practical application. And again, MIT, of course, being MIT's technology, MIT was just poised. So it took a long time in neuroscience. MIT was very slow. But in general, in spirit, MIT, I think, has always been ready, willing, wanting to lead in turning some piece, some intellectual pursuit to some output. Translational is now what it's called. So that spirit, an entrepreneurial spirit, and you could call it partly, but not entirely, that's been around here. But bit by bit, as I say, that's liberating, or at least it makes you think, well, gee, maybe we could ask, what is the nature of thought? And then bang, along came yet more methods in imaging and so on. So it's a great time now in this field. It's just awesome. It's wide open. It's just open, And I can't tell you, I don't think anybody knows how the brain works. I don't think anybody, I don't care who you talk to. I personally wouldn't believe them. We don't even know the frequency of the signals that are important. I mean that literally. And 10 years ago I would have wondered, what does that sentence mean? But what it means is that we've become used to thinking that the main activity of the brain that's important are these little spikes, little digital spikes. But we've always known, or we've known from the last century in the 30s, that there are these waves, and oscillations in the brain. They're like EEG and sleep wakefulness patterns and so on. And AI people have been interested in them for a long time. But most people just kind of set that aside. Well, now they're hot as fire, because maybe there's another way the brain is signaling to itself, one part to another by kind of like a carrier frequency, and then maybe one of these little abrupt little spikes, if it hits a peak in the carrier frequency, it's more potent. We don't know whether that's true, it's an idea, but it just shows. Or let me give you one more example. So a long debate in neuroscience from way, way, way back when we knew almost nothing. In fact, a classic debate was whether the nervous system is a syncytium, whether all the cells are physically continuous with one another through some little strand of cytoplasm, or whether they're discrete entities and their only interactions are through the so-called synapse, where electrical impulse comes down to the end, chemistry occurs, molecules are shipped out into this space, the next cell receives the molecules by adapter molecules, and more chemistry occurs, and so on, and more electricity. Well, now it turns out that probably a lot of the communication goes on not in the synapses, but maybe near them, but like not in them. Well, that's got to be a totally different kind of communication. Again, it's going to be more punctate, more digital or more analog, and on I could go. People are debating, is the vasculature important for the brain's work? Because all these imaging studies are based on looking at blood flow. You know, they're not looking at what the brain does, looking at the way the blood flows. So this is all part of the freedom that, at least freedom to fiddle around and freedom to think that maybe you could solve anything. You know?
Are those stories and the focus on the problem that you described as being coached into basic research, look at that problem and see what comes out, right. Does that in any way mirror or remind you of the way that your dad particularly approached nature and the ways that he would explain to you what was going on? Is there a sort of a continuity in approach to science that goes back to his training?
Well, see, he was a physician as well as scientist, and so I think he represented both sides, both the pure and the applied. And he got interested in, he worked on the heart. He worked here in Boston, the MGH. But then in the war, was sent to Florida, and that's where I grew up. And down there, he found that, or it was found, that pilots were crashing. And my father had worked at the Harvard fatigue lab. They figured the pilots were tired, so they said, please solve the fatigue problem. But it turned out they were disoriented because of their vestibular systems were being stimulated in unusual ways due to changes in gravitational levels as they flew around. Mm-hmm. And so he ended up doing a lot of work on the vestibular system, which led to a lot of work in the space program. So one of the fun things I got to do when I was little was to go to Cape Canaveral, and I was there when John Glenn lifted off, and I was kept, again, it's a woman's thing. I was the daughter of the physician to them, so I stayed with the wives, but it was very exciting. It was wonderful. It was wonderful.
That's amazing.
Stay up all night and then it doesn't go. Stay up all night and it doesn't go, day after day. And
you didn't want to be an astronaut, though?
There weren't any, well, I mean, stuff like that never came to my mind. You know, that's what, maybe I'm just backward or something, but women weren't astronauts, you know what I mean? It just, so I'm afraid, so no, I never wanted to be an astronaut.
Again, I'm not holding that against you, or the not burning your bra thing. That's perfectly okay with me. You know, it's just interesting, this journey from one way of looking at things to seeing the world as completely open as you describe it. I mean, in the course of 2 minutes, we've gone from there was one way of looking at things to also saying it's completely wide open if you're thinking about the
Yeah, and this is just, I mean, this is so old hat because now everything is open. That's what I love about now, you know. Mm-hmm. I mean, internet, you know, YouTube, the whole bit. It's just, it's fabulous. So I mean, I guess it's very good historically to document all of this, but it's going to seem so bizarre. I mean, anything that you and I are talking about to anybody who's under, you know, I don't know what age, but it's just unimaginable, isn't it? Mm. Right?
How did you find your way to MIT?
So I had decided to go to Tufts because I was working on a project as an undergrad, and I wanted to follow a project. And then I heard about MIT. I heard they were working on the brain, and I was extremely, by then, passionately interested in learning about brain and behavior. In fact, at Harvard, I tried to get them to let me do a combined major in biology and psychology, but they wouldn't let me do it. Now everybody does that, but
Which did you choose?
Biology, yeah, right.
Why not psychology?
It was very soft at that point. It was, but I heard fantastic people. George Miller and, Jerry Bruner and Roger Brown and many people like that. But there were, I was pretty busy. I took a lot of, in those days, we had labs, I had a lab every afternoon. So by the time you got done with classes all morning in labs, there wasn't a lot of time. But, so I heard about Teuber, this phenomenal guy who had this young department, and I started going to the colloquia here on Fridays, and I don't know whether you've ever heard of them because you visited, but they were awesome. People from all over came here on Friday afternoons to a little room in a little ex-automobile factory building to hear these people speak. He had everybody. He gave long historical introductions about the field and about the person, and so it became clear this was pretty interesting. But I had thought I might study insect nervous system or at least invertebrate, somewhere in there, because it might be a simpler situation, might be able to solve something. And so I got in here twice actually, but I didn't go. I stayed at Tufts for a while, but I'm very glad I came. But it was just the force of what that fellow did, and it was. MIT had been so strong in physics and math, and as I was trying to say earlier, there was brewing this fantastic group of people interested in the brain and in computation, really. But they were quite separate from the people who worked on the brain or in psychology. So it was good to have the psychology department, but then Jerry Litvin, he and I have done experiments together. Again, an older person being awfully nice to a little one. But these guys they're fabulous people. So I learned a lot through them. And Walter Rosenblith, I hold the chair in his name is tremendous honor. These are people who really thought about what is the essence of communication. And I, for a long time, have thought of the brain as a communication system. I was caught up short by somebody from the old Bell Labs saying, no, the brain is not a communication system, but a lot of communication goes on in there. And again, this very attractive about MIT, this blending of a more formal computational or, if possible, mathematical approach, and then biology. I'm sure it's a way to do it. Not sure exactly how, but put a lot of smart people together, and I think actually that's what's going on now.
A lot of people who conducted brain research did choose to go the invertebrate route. Candell,
Right.
finding giant neurons and sea
Right.
slugs and those kinds of things.
Right.
You chose to go straight at the human brain.
I was very worried about doing that because I was very lucky. I was exposed to a man named Kenneth Roeder, a German. I would say that 3/4 of people to whom I was influenced or exposed and became influenced by them were Europeans of one sort or another. And this man, he had discovered that the moth has 2 sense cells for hearing, just 2. And he tried to go from those 2 cells into the thicket. The invertebrate nervous system it may look nice in a picture, but it's really actually very hard to work with. And then you go out and, the moth will either come avoid a bat or, go toward a mate. So he was trying to solve that problem and had the idea of this extreme simplicity would help. And I just almost did it. But the human brain, is such a mystery. And, I don't know about you, studying gravity would be another big one, or in general studying the universe would maybe that's even better than studying the brain. But the brain, I would put those almost on a level in terms of interesting and very, very important things to study. So it became clear. So why didn't I go right on to brain of human? There was no adequate method. But I had learned anatomy here, and I was interested in chemistry, and it dawned on me that if you could see the distribution of the chemicals in the brain, you know what you could do? You could go from an experimental animal to a human brain. And so that's what I set out to do, and I actually did it for some time. I would do an experiment, find something in the animal brain, try to figure out a neurotransmitter that might mark that phenomenon, and then get a human brain and try to get the same stain to work, which would then, you know, it's indirect, but it's better than nothing. So that was an amazing time, because there were no rules, and so I was just this person in MIT. I would go over, it's amazing, I would go over to the MGH. I found out where the deaner was, and I went, and I asked if I could have a human brain, and pretty soon we became friends. And I went over there a fair amount and was able to obtain very, very, very fresh human brain specimens, and that's where I began to see. I saw autopsies and, you know, did my dutiful fainting spell stuff. But that was really incredible, because we did find some things in human brain that I'm very much hoping will be useful in human, you know, trying to help human disorders of certain parts of the brain. So there wasn't a way to actually go all the way then. Now, I don't know now how far human brain imaging is going to take us, but we're only at the very first wave of that. So I think we're there. We're just there. So
eventually your research began to focus more narrowly on a particular location in the brain that was as mysterious as everything else, but possibly narrowed the problem for you, simplified it, made it more manageable. How did you come to fall in love with the basal ganglia?
How could anybody not? You know?
Yeah, I know, it's like certainly.
You know what I mean?
Yeah.
I got it bad. I really got it bad. It was this, I've been working on sensory motor things, and I've gotten closer. I've always liked to be in the middle, like not fully sensory, not fully motor in what I study. So I had always before worked kind of in the middle, but in the sensory motor middle. And then I found some things that actually implicated the basal ganglia, or some pathways related to the basal ganglia, in the control of eye movements. That was interesting. And meanwhile, I'd been trying to get this, can you use kind of chemical neuroanatomy, which didn't exist, to get at the human brain. So I did around then the dopamine had been found to be implicated in that transmitter in Parkinson's disease, right? So the big idea then was that dopamine is fought by acetylcholine. So they're in a balance. If you have too much of one, you get one thing. If you have too much of the other, you get the other thing. For example, schizophrenia or Parkinson's. So I went after acetylcholine and stained human brain and a ton of different, the animal kingdom of brains for cholinergic markers. And it was very exciting, because, see, our concept of brain has been, and still is, that we have this outer rind, the neocortex, which is the stuff of higher cognition. That's where we do our math. That's all the high-level stuff goes on out in that outer rind. And then deep down, underneath that, either things are serving the cortex to prepare for action, or they're serving the cortex to prepare the information so that this cognitive machine can take over. And what was discovered when I was a student was this beautiful cortex has a magnificent architecture. It's divided up not only into layers, it's been known for 100 years, but little columns like Greek columns, and at the intersections important things went on, and you could have columns that saw things from one hemifield, or the right eye and the left eye, and so on. So what we found down in the deep brain was sort of like that. It was in effect that, my goodness, the deep brain isn't just a homogeneous little ball of low-level cells. There actually is a magnificent architecture. It's just nobody had ever seen it before because they didn't use these chemicals. So that was a trick, you know, to see it. And then once we saw it, it was, our lab, many other labs started staining for everything. And it turns out that just about every molecule we've got in there, and there are a lot of them, are divided into this kind of architecture. So that was exciting. And then the hooker was that we still had, it's deep brain. So it's hard to get the microelectrodes in there. You can't see this part with any method. This architecture is too small to this day to be seen with fMRI.
Really?
Yeah, it's really, it's, but we're almost there. So we are, man, would I like to do that? But we're trying, we're trying to push it as far as we can, as fast as we can. But then it turned out that, again, MIT is so special. A lot of computational people were interested in learning architectures, okay? And that's what I meant by, that's one way to go at what the brain does. And we saw connectivity patterns in relation to this architecture that for all the world looked like certain computational models, at least in their schematized views. So I talked with Mike Jordan and some other people, and I completely fell for it and decided that the striatum in the basal ganglia, big input side, great big structure, huge in the human. This is a learning, maybe it's a learning machine. And I actually still think that, but that then meant we had to change. We could not simply know where there was this architecture. We had somehow to get in there and get at what it does. And the way to do that, the only way that we really have so far is with microelectrodes or doing imaging, which can give you an idea of function, but without this fine detail that may be the catch the whole thing.
The trajectory of your research was, first of all, you found this complexity, this anatomical complexity, which suggested this is important. Then you began to look at the disposition of the chemistry,
right,
and began to see patterns that suddenly were familiar to some of the computational people across campus.
Right.
You began to discuss whether that similarity was real. A theory emerged about
right,
this sort of communication learning system. Right. And now you're just getting to the point where you may be able to actually experimentally, on a broad basis, test that theory.
Right. So let me just say this, instead of narrowing, this all started with a question of, well, you've narrowed down. No, it turns out that when you go deep, you go broad. And so first we've had to develop and borrow these multiple microelectrode methods. It's very exciting. You try to get as many electrodes in as you can to record simultaneously from lots of neurons and record over a lot of different frequency ranges. So you get all this, digital stuff and all the oscillatory activity. And because of the learning, and because of, the basal ganglia are also, I must interrupt myself, the basal ganglia are also interesting, as I hope we can talk more, because on the one hand, it is turning out, as I've learned more, and as the field has, that the basal ganglia are mixed up in many human disorders.
Well, let's list some of those.
Well, there are the ones most people know about, the kind of movement plus disorders, so Parkinson's, Huntington's disease, dystonia are the three great ones. And then neuropsychiatric disorders, obsessive compulsive disorder, Tourette, ADHD or ADD. autism, maybe depression, probably depression, and in unmedicated schizophrenia, maybe, you know, we're going down the list of certainty now. But in other words, not only neurological disorders that have to do with movement control, which basal ganglia used to be thought of as one half of the movement control system that isn't the cortical control system, which is the kingpin, right? So the pyramidal tract, as you well know, controls the spinal cord and so on. But then there are two great brain systems, the cerebellum and the basal ganglia, that help that. Well, they help it, yeah, but they may help it in ways that have to do with plasticity, which is so key to any kind of action. And so for the basal ganglia, what's really happened is that the one whole side of basal ganglia work has blossomed as more and more it's been realized that, well, I mean, they are in these loops with the cortex. I haven't told you what they're like, but the cortex and basal ganglia are constantly communicating with one another. It's just a set of massively connected circuits, and the one influencing the other. And so these now we would accept, I think the whole field would, are involved in these kinds of disorders, bipolar probably as well. So then the other part of the field, which was ignored for a long time, there were psychologists, old-fashioned psychologists, saying that if they made lesions in the basal ganglia, then the animals had trouble with something they call habit learning or learning mazes or something like that. And so a couple of really amazing things have happened. One is that people discovered, not at MIT at all, that this dopamine is so important for Parkinson's and so on, that it is a transmitter in a system that basically responds either to primary rewards like chocolate or to some conditioned stimulus that represents a reward, or to a predictor of a reward, or maybe to, and is, that signaling is influenced by how certain one is about a reward. So it's become so now we know that the place I work on, which is heavily influenced by this dopamine system, for example, will light up in fMRI studies when someone sees a picture of someone they're in love with, as opposed to someone they're just friends with. And so there's a whole side of that now we can appreciate the basal ganglia is somehow very sensitive to, maybe it's reward and punishment, most people think mainly reward, but somehow to reinforcement. So you could imagine a system that, you know, if you're constantly monitoring and then kind of adjusting the gain on pathways that help you decide what you're going to do and whether you're going to do it again or which path you're going to choose, you know, at a decision point, that if that happened over and over and over, then you might develop a habit or a pattern. And that's become quite a central focus of what we do now. And it's unbelievably closely related to these clinical conditions, which is what is, it's seriously exciting. It's just, it's very, very exciting.
Addiction is suggested.
Yeah,
right?
so what happened with me is it went from these chemical architecture stuff. I figured it might be a learning thing, architecture. I found that was generalizable to not just the neurochemically identifiable parts of this system, which was a big step up. And then I had heard that someone in England working on the spinal cord had made a stimulation that would excite the spinal cord, and he had used a method to look at very rapidly activated genes. They're very rapidly activated when the cell surface is stimulated on even a single cell. And that was the beginning of a method that now is used by many people, but this fellow had been early on doing it, and I just happened to see it and thought, oh my goodness, So I tried. We gave an animal a rat, a shot of amphetamine, and we chopped up the brain and did more of this chemical anatomy type visualization, but this time of genes. And darned if we didn't find these compartments that we had discovered all those years before. So that was exciting. So that began a line of work it's curious. Things are coming together. I guess live a long time or something. So that was absolutely cool. So then, which I knew nothing about drugs. I'm no expert, but you know, obviously you get addicted if you're exposed over and over, and there are beautiful routines for studying that. So we picked up those and did this to animals, and we've now found in mice and rats and monkeys that if an animal is given this drug more and more and more, cocaine, amphetamine, we've used a number of different ones, that this pattern, which emphasizes the stuff we discovered way on back, that pattern, that emphasis gets greater and greater. Well, that's interesting. But then I was very lucky because a fellow came to the lab as a postdoc, and he had studied what behaviors are induced when animals are given such drugs. And so we did more experiments to see whether, when you give these drugs, of course, the animals, they get hyper, they develop what are called stereotypies, or repetitive behaviors. So little rat may rear over and over endlessly or chew on something endlessly, and monkeys do it, we do it, we all do it. And in experiment after experiment, how repetitive, how stereotypic the animals were seemed to be correlated quite amazingly closely to how much this pattern was emphasized, the striasomes. And so that was interesting.
Wow.
So suddenly we had a situation where, see, I had been trying to stain human brains right along, and we still have not, well, anyway, I don't want to get too sidetracked, but it became apparent that, so we did some more old-fashioned track tracing. The last I did way back, a long time ago, we wanted to know what parts of the cortex in a monkey would preferentially target these compartments that we called striatal bodies or striasomes. It turned out, of all things, the only two places we found were the two places that are especially activated in drug addicts on the one hand, and in people with OCD. So they have abnormal activity in this far anterior cingulate cortex, caudal orbital frontal, and the striatum, especially the head end, where striasomes are, these compartments are very big in the human. So, you know, it all, you know, it's very enticing. You know, all of it may just fall apart and it's just a dead end, but maybe, I hope it's not.
But
so
it does seem as though patterns of movement and patterns of behavior are localized in the basal ganglia to some extent, and the brain appears to consider them, at least at that level, equivalent. They're just different pieces of information.
That's right. I think patterns of thought and patterns of movement are controlled probably in much the same way. And exactly what you said. So apropos what you said, when we have not yet, with microelectrodes, been able to find these compartments, and we're trying as hard as we can, and I got some wonderful new people. We actually have I think a real, I don't know, we're all excited about trying again. But what we did find, we did this very simply. It's just what you're talking about. We put rats in a T-maze, just like psychologists use, and they have to, a little door opens and they run down this maze, and then they either feel one of two textures under their feet, or they hear one of two sounds through loudspeakers. And depending on what the cue is, they should turn either right or turn left to get a reward. And we literally just put electrodes in the sensory motor striatum of these animals and did that chronically as they learned. And very oddly enough, it's so hard to do. It's not anymore, but it was so hard to do that very few people had done that chronic day after day recording, you know, take them, bring them up, take them to school every day and record. And I have to tell you, what we found was absolutely amazing. And we found it over, I know it's true. I think it's true. So when they start out and they're running, and they do like this 40 times every day. The cells in the striatum, the nerve cells, are firing on average kind of all through those runs. They just, as though everything is interesting and everything is, important, or somehow they're going to respond to just about everything. Not every cell, but on average, take the whole population. Try to record from thousands, okay? And then as they learn, the activity, right when they start out and right when they get to the end, it's very strong. And the activity in the middle gets weaker and weaker. So it's like a get it going, and then don't worry about it, and then get it stopped or get to the reward. It's as though you bracket the whole behavior. It's exactly what you said. And so then I began to think, well, maybe one of the things that basal ganglia do for the cortex is they help decide if this is looking good, I get rewarded when I do this, so I want to do this more. The basal ganglia help whole behaviors get kind of packaged or chunked, and then they ship the information off. Then the cortex is the grandmaster and can, execute it however the cortex wants to do that. But this guy is constantly kind of helping make more probable one or another whole stream of behavior or a whole stream of thought. And so, if you think of it that way, that's interesting. So we're trying very hard to track down what is it exactly that makes that pattern happen. And we're doing that in different kinds of animals.
Your lab has a lot of undergraduates. Is that because, well, what is that because? You're known as a fairly popular lab on campus.
Oh, good. I didn't know that. I'm very happy. So, well, they're great kids, right? So, I mean, I actually have been told that, it's stupid that we do that because it takes some time. But I always have done it, as a matter of fact, and partly it's, I just don't want to sound I don't want to sound the way I don't want to sound. I don't want to say that, it's really nice to help people, but it really is nice to help people, and that's the way my folks were. But it's more than that. It's that, some kid comes in and he's just fascinated, gee, I'm interested in the brain or something like that. And so why can't they at least get exposed to it? And I will say we've had a lot of girls, because that's one of the ways I've tried to help. So I've had even high school, had a fair number of high school girls, and when I was very young, the lab was really silly because we had different aged girls who, because see, I told you I was never exposed to any of that except through the privilege of my particular advantaged upbringing, but why not let them see what it's like? And so in the end, I was so surprised they had to get me to tote up all the Europe's, and I guess we have had a lot of undergraduates and a lot of women undergraduates in the lab, and I can't believe them. I mean, they're all beautiful, they're all brilliant, and they're all able, and almost every single one of them comes in and does something just fantastic. It's really great. So I love doing that. I used to do it all by myself, but now, it's only in the last little bit, we kind of like one just works with one person sort of thing. That's the way labs usually do it, but I used to do it with them myself. But I can't say I'm actually working with an undergraduate, but I talk with them, and I think it's just, it's when I was an undergraduate, there was very, very little of that, but it was just a pure fluke. But the year I went to Harvard, they started the freshman seminar program, which is a laugher at MIT because, everybody does that sort of thing, but not at Harvard. And so I was accepted into the freshman seminar program in chemistry, and it was bizarre because I'd never had chemistry. I didn't know chemistry from Adam. And
Home ec, really? There's a little chemistry.
yeah, exactly. Yeah, I love to cook. But there was this wonderful man, Eugene Rockwell, bless his soul, and he took me in. Everybody else, I think I was the only girl, I'm sure I must have been, but they all like, you know, basement chemistry setups, and then I'd never even seen a crystal. I was dazzled. Oh, I loved it. I just adored it. Just so, maybe that's also part of it. It's really great to let them get excited, you know.
Young, undergraduate, young.
Yeah,
women,
yeah, but
yeah.
I mean, any student.
So you said something on, like, it took you a really long time to feel as though it was okay that you were a woman doing science. Maybe you misspoke, but was there a point after a while when you looked back and said, oh, I'm comfortable with this now, and maybe I wasn't so comfortable with it earlier on?
Well, I don't want to paint a one-sided picture. I mean, you know, how can I put this? It also can be fun to be the only woman in a, you know, I had a lot of fun at a lot of different occasions. No, it's more that all I meant to say really was that it was unusual to see, especially when I was an undergrad, and then even here, there are a lot more men in the faculty than women when I came. No, I mean, I've always felt as though I could do that, but.
When did you notice it wasn't unusual anymore?
It's very field dependent. I mean, it's still unusual, but I'm amazed that I can, in math at MIT, there's so many women. It's just great. I just found that out. But I don't know. I've never, I'm sorry, I haven't kind of made being a woman kind of like the main thing in my identity. I've been pretty interested in just doing whatever I was doing.
Were you, you were...
Except trying to, I feel very strong, this business of helping the young ones. Because I'm sorry to interrupt you, but my view is that certainly with me, but I think with many, many of the women, at least in my era, and I still see it, is that they don't really have that fundamental self-confidence, you know, I can do anything kind of attitude. And so I try to, the ones who need it, I try to give it to them. Because I think that's, now that is empowering. Mm-hmm. If you just, and I think a lot of women are to this day timid, at least in some fields.
Were you conflicted when Nancy Hopkins came to you, wanting to know, first of all, your experiences, and secondly, if you'd participate in this report?
No, I think it's terrific what she did. I think she did an amazing job, and I was one of that group. Oh no, I mean, I think she, you know, she's had women all over the world come to her and write to her and invite her and so on, but I've had a fair number of women myself all over the world who've come to, you know, how do you guys do it, and can you really do it? And I think it's magnificent that she was able to, it's awesome what she did actually. We never even knew. I mean, I didn't know that I was that underpaid, or I knew certain things about space, but you know, but she did a lot of good.
How did the institution of MIT either hinder or help the process of solving this problem of gender equity, and how has it changed the institution?
I think, you know, honestly speaking, MIT has come around. I think Vest, ex-President Vest, Chuck, really embraced the issue, kind of said this was one of the main achievements of his administration. So I think MIT's tried very hard, and now, I mean, we have Susan, so that's very good. It takes time to, you know, it does take time, because you have to find enough people to fill the slots. But I don't know. I think it's okay. That I think it's going to take a little more time. I'm afraid I'm cynical about something. We're going to have to see. I'd like to see how many people, how many women starting out now stick in the field and, you know, become accomplished professors or whatever, you know, business women or something after a while. But gee, the Institute has changed radically. You know, there are huge numbers of women now, and they're as I said, they're doing wonderfully. And they don't seem, you know, depressed and, you know, working all day and all night and sitting in a corner, you know. They seem like they're out doing all kinds of other stuff too. It's just great. And
Nancy describes that it was fairly clear to her that to have a life in science, she would have to forego other aspects of life.
Right.
Did you see that trade-off?
No, well, in one way, yes. I mean, it took a long time to decide about children. I was scared that I couldn't do both. But we wanted children. We didn't have children, but we sure wanted children. I very much wanted children. Didn't have any. But I'm happily married to the same guy, an MIT undergrad himself. And I don't, and so I have, another thing my lab became noted for was the number of children born to members of the lab, certainly including almost every woman. And so we have a rogues gallery of kitty pictures, and the water down the hall was called fertility water, all of those things. So I think why shouldn't people be able to, you know, manage all this? And the actual, I mean, the thing is that for families where they both try to help, and especially if they, I don't know, it's still difficult. It is, I mean, Nancy's got a point, and I think anybody who, again, it's field dependent. If you don't have to be in a lab, if you can sit with a laptop, do math, you know, something like that, then I would think it's a easily doable thing. If you're really a research scientist, you have to be in a lab, then it's difficult. But women are more and more doing it, right? Their husbands are more accommodating. Maybe we're getting better child care. I mean, that's one of the things MIT's trying to do, has done. That's great. I mean, I know women who work in the building I'm in, and yeah, they pop their child in the daycare center in the next building, you know, can visit the child at noon, pick it up at 5, and that's it. So, oh, it's tough, but a lot of things are tough.
Is it personally vindicating to your choice of study and the big questions that you decided to embrace that the president of the university is a brain scientist?
Is it personally vindicating? I celebrate it. I think it's just wonderful. Personally vindicating, no. But she's fabulous. And
certainly biology is not as much of a fringe as it was when you first came here.
No. So I guess I didn't emphasize that enough. I mean, the changes at MIT are really are profound. It was a place for math and physics, engineering, architecture, and so on. And then biology became so successful. And the new wave of biology is neurobiology, right, and neuroscience more in general. And so if it's a vindication of anything that she's here, it's a vindication of the success of that field, and then her own incredible ability. This is one talented lady. So that was a good move, and it to me anyway, it seems very natural. But do I notice it every time? That wow, you know, there she is. Of course I do. But I think it's great. But you know, it's true everywhere, right? I mean, it's true at Harvard, maybe true in the United States.
Right. How important was your relationship with Walter Rosenblith?
Oh, he was, as I said, I'm so honored to have that chair. He was the key man thinking about the brain as a communication system. This sort of Sensory Communication was a book he edited. It was very famous at the time. And so he sort of stood, in my mind, for that hard more engineering or computational side of things early on, and signal analysis. And then as he and Jerry Wiesner came to run the place, they were terrific to me. They had trotted me out to talk a lot, so I gave a number of talks, and Walter was always very funny and teased me a lot, so I thought I enjoyed him personally. He's a very, very talented man. But he was not kind of among the group in the department that led to this neurobiology part. He was the other side, and now the two have come together, which is almost come together, coming together. So he was an important man.
You won the National Medal of Science in 2002. You're also the recipient of the Killian Award. You know, aside from 2002 being an exciting year, how do you evaluate your recognition, partially in those awards, but in terms of the field, what is it about your work that you think is worthy of recognition, that answers a specific question, that is a contribution, that is clear and discrete?
Clear and discrete. So I told you we discovered some things, and that is ongoing with the gene work we're doing. Tell you the truth, I was absolutely amazed at each award, and beats me, you know.
Oh, shucks. Florida Panhandle coming right out there.
Yeah, I mean, I'm very lucky. So it's, I haven't solved. I'm on the way though. I'm just, I'm very, very excited about what's going on right now. So we've added, I mean, there's no doubt, our lab has added something to this. We've added a brick, right? But there are very few single discoveries, we all know that, right, which completely transform a field. I've not, to my knowledge, made such a discovery. However, I am extremely interested in whether the relation between this kind of architecture and some of these human disorders can come together, and that may not be so much a, I don't know how you'd put it in terms of a discovery, but it might be very helpful. So we're hard at work. That's where we're going now. I like what we do. I've now embraced the idea of that pure science can be applied science. That's exactly what it can be. That's my new idea. And so that's what we're gunning for. I want to get, you know, some of these disorders. I'd like to, we're working on genes, we're working on all these things I've told you about, seeing if we can really hit something that will help.
Do you think these fast transforming genes are a key to understanding a whole new mechanism of communication in the brain?
Why not? Yes, I do. And, you know, that's a general idea by now in the field, that there's a whole cascade of genes that act in different time domains, and the ones can set off others and that can then go back and influence the ones. Biology is multiple control systems, one on the other. It's just, it really is. It's profoundly, it's, again, awesome. It is awe-inducing. Yeah, and actually one of the little things we found they're pretty interested in now at the Broad, one of those early genes, but we have now found, I decided that if we had to clone genes, it's funny coming in my lab, not being raised as a molecular biologist, but I did go to school and learn some. And we now, in collaboration with some terrific people, David Hausman is one, we've cloned some genes. We wanted them selected for this, the basal ganglia and the striatum, and gee, we've hit on some real doozies, I hope. So they turn out they're expressed, again, in this compartmental way. And amazingly enough, if you knock them out, which a wonderful young woman in the lab is doing, so you make these knockout mice, they have abnormal stereotypic behavior or perseverative behaviors. So, you know, can we help in autism? I don't know. Can we help in OCD? I don't know. But we are really at it. And the other funny thing, it's funny how these things are all, it's just as though I just have a wonderful sense that things are coming together, because these two that I'm particularly talking about, they're regulated oppositely in Parkinson's models where, you know, in people, if you give them levodopa therapy, Cinemat or that kind of, then a lot of the people, unfortunately, develop abnormal movement patterns called dyskinesias. So you'll see a Parkinson go from unable to move to waking up to, you know, having all kinds of choreoathetotic-like movements called dyskinesias. They're awful. They're the biggest problem in therapy. And in a dyskinesia model, the amount our genes are regulated, the two of them, each one of them actually, is correlated with the amount of dyskinesia, which is perilously close to the amount of stereotypy related to, you know, them.
So it's trigger meets trigger meets trigger.
Yeah, and then we find in Huntington's brains, I actually worked on the brains of Huntington's patients, these compartments are differentially affected depending on whether the major symptom is a mood or a motor symptom. And in models of Parkinson's now, we're just beginning to see these genes now differentially regulated. So, and now the bipolar community is interested in this. So to my utter amazement, we're giving animals lithium. I mean, it's just, this field is bubbling, you know. It's just bubbling. And so I'm very, I mean, there is a sense in which, aren't we lucky, people who get to do this kind of work? I mean, what I want to do now, I want to help people. And that my tools, my ammo is to do this kind of work. And I don't know whether we'll get there, but so many things are coming together that I sort of think if we don't, you know, as one of these nice girls, well, it's great. So.
Is that what's kept you at MIT all these years?
That's strange you ask. I have a terrific guy in the lab who made that picture, and he said, so you're going to go get interviewed. The central question is, what has kept you at MIT? And I hadn't really thought of that, but I have been offered some other places I might, as you can imagine, but if for no other reason than they needed a woman. But MIT is just too cool. It's really, what is the essence of MIT? It is a place where you can go ahead and be smart. You know, you can go ahead I think for a lot of people, go ahead and be you, and just kind of go for it, you know. I had a kid who came in the lab and said, gee, I have an idea, you know. I want to make a better robotic wheelchair, and then people are doing that, so I want to get it in an automobile, and then he's this little kid, he's running rats in our mazes, and he's thinking these thoughts, and I could pick up the phone and say, why don't you go see, the head of the instrumentation lab and find out what they're doing. This is MIT, but so is music. I mean, MIT is a very interesting place. So I think it attracts people who are smart. A lot of them are smart. A lot of us are rough diamonds, but that's okay. That's okay. So I think there's a sense of energy at MIT. Something's always going on, you know, and a sense of excellence, a kind of expectation of excellence. And of course, every university aspires to that, I'm sure, you know. But this is a kind of high concentration of that kind of person. And it makes it interesting. We're all a little bit odd, right? Or at least a lot of us. I would certainly include myself. But that makes for being interesting.
Is there a way to answer the question, what is MIT's role in the world?
Well, its role is always going to be changing, right? But I think this, it's nice to have a few places in the world that do have that set of standards and so openly declare it. You know, you can think anything you want here. You can do anything, right? You can put a car on top of the dome. You know, literally, you can do just about anything. And so I think that attitude put in the context of some intellectual framework, I'm big on, you know, pushing the intellect. I think we don't use our brains anything like, that's the big question, why we don't. Why aren't we more motivated, right? So I think part of what's good about MIT is it keeps a very high level of motivation in its people, and I'd like to see that spread. Like that's a giant problem with human beings, and it brings about a lot of our difficulties. And it's, you know, of course, I always can relate that to what we do, because there is this apparent disconnect between what we think and want and intend, and, you know, I promise today I'll be different or better or whatever, and then what actually comes out of us. And I don't know the degree to which it's different parts of the brain unable to talk to each other. You know, why is it that some people work feverishly? You know, why is it? I can't believe it's just that they're smarter than other people, right? Or some people are passionate to paint, passionate to write music, you know, or whatever, right? Play the banjo, you name it. Why? And then other people just they just can hardly get through a day. And wouldn't it be wonderful to, you know, have them come up, have them experience more of what the other people are experiencing? And so MIT, it represents that in a very real sense. And I think it's great for kids to say, gee, could I ever get in MIT? I think it's wonderful. So.
You know, you've enumerated in the course of this hour and a half probably about 30 or 40 fairly big questions about the brain, how it works, what is thought. Is there one question in particular that you would love to have a part of answering that is one of the big Anne Graybiel questions about the brain?
Anyone would be fine, thank you. But I think this one I just was talking about, that we are, there are disconnects between some level of thinking and planning and deciding, disconnects between some of that and some of this deep core stuff that actually ends up driving our behavior, right? So I don't think, how many people do you know who actually do what they think they ought to do or want to do or, you know, decide to do? How many people like that do you know? You know? So I have I'm feeling probably shared by many people in brain work that there may be just small little adjustments that we need to make in either motivational systems or somewhere in those little neurotransmitters that are connecting the cortex with other parts of the brain. Just minor little changes that would have the machine kind of work optimally. And although some aspects of our machine and our brain do work optimally, you know, it's amazing. We're actually studying that. It's amazing that you can put a monkey to a little task that he has to learn all by himself. You never teach him a thing, not anything. And he will learn that simple little task, ours is an eye movement task, to what a high-level physicist, computational guy, my colleague, tells me is the optimal solution. The brain just does it. So Norbert Wiener said that, you know, any early learning, all early learning is a miracle. And it's a miracle that we can get these different aspects of brain controlling behavior to gel as well as they do. But so I'd like to up the ante. I'd like to get them to work together just a little bit better.
You know, everybody knows how competitive people are at MIT.
Tell me about it.
The sense of...
I know also.
Let's hear what?
I also know, yeah.
Yeah, I'm sure you know.
Yeah.
If there was a competition between the people who experienced the first 50 years of MIT, the second 50 years of MIT, and the third 50 years of MIT, I suspect you would say that you're 50, you're coming in this third period of the 150 years is the best.
Ooh, best. What do you mean best? They're so different. I mean, war years, right? MIT was central and crucial to the whole United States effort, and right, for a lot of years, right? MIT's been crucial all along in so many different ways. But
most exciting 50 years in those 150 years.
Man, I wonder.
You got to make a choice.
What would a physicist tell you? He'd say, "My God, the physics department was huge, and it got to be tiny, you know, and now we're trying to get it to grow again. These are the bad years."
For the physicists.
Yeah.
But what about
So,
hands?
well, you said for all of MIT.
Well, what
so
about
it depends
for
on the part of but I mean, hands down, for anything to do with studying biology and brain, it's just razzle dazzle. This is the time, man. Go for it.